This is lesson 10. What do cells need to grow and make more cells? So we've seen that cells can make more cells of the same type, but we need to know, what do they need to be able to make more cells, and where do they get the stuff they need to make more cells? So we're going to compare bacteria cells to human cells. So what do you notice about bacteria cells compared to human cells? All viewed at high magnification. So on the left, we have two different types of bacteria cells, and on the right, we have the three major types of human cells that we've been looking at. So think about what you notice that is similar between all the cells, and what do you notice that is different? So the type of bacteria cell we're going to focus on here is the E. coli bacteria. So that's the picture up top. Scientists use E. coli because they are easy to use in the lab. They are non-disease causing versions or strains, so there's types of E. coli that won't make us sick. And they share some similar features with other cells. So scientists, when they want to study things like bacteria, use a gel called agar in which to grow cells in the lab. So what the heck is agar? Agar is a dry powder made from the red algae commonly found throughout the world's oceans. So this first picture shows our algae. As a powder, it dissolves in boiling water, and when an agar solution cools, it forms a semi-solid jelly. It's like an extra thick jello, basically. When scientists create agar plates by pouring hot agar solution into a petri dish and letting it cool, they can then have a source to grow bacteria on. So when scientists prepare agar, they can add various amounts of food and nutrients in different agar plates. So how would using different amounts of food and nutrients help us figure out whether or not cells need food to grow? So stop and answer that question. How would using different amounts of food and nutrients in these agar plates help us figure out whether or not cells need food to grow? What kind of experiment could I do? So we're going to test the question of how will bacteria respond to different nutrient levels. We want to be able to give our bacteria some food to see if they make more of themselves. Scientists can add food and other nutrients to those agar plates. So our first step is going to be to prepare solutions of agar with different levels of nutrients. So if this is an experiment, this is my independent variable. I'm changing the amount of nutrients in the agar. So if I'm going to grow my bacteria, I have to do something called streaking. This is adding the bacteria to the slide or to the dish. So you first sterilize the little loop. We don't want to add anything beyond what we want onto the plate. And then let it cool a little bit. We also don't want to cook our bacteria. Okay, this container contains the E. coli. So we're just going to dip the loop into the E. coli, cap it back up. And then this is streaking. So we're going to just rub the bacteria back and forth over the top of the plate and then sterilize our loop again. All right, next step, we have to incubate the plates. So that means putting them at a nice warm temperature to let the bacteria grow. And you'll notice the temperature is 98.6 degrees Fahrenheit. That's body temperature. So we're looking at how things would grow in our own bodies. And they're then left in the incubator for 2 days without being disturbed. So this incubator basically maintains a constant temperature for our bacteria. All right, in your packet, you want to open it up to page 12. On page 12, you're going to answer the two questions that are there. The first question, when the bacteria are on the agar plate with no nutrients, So it's just agar, no food or nutrients added. What do you predict we will observe on the plate? So will there be a change in the bacteria if I put it on the plate for 2 days with no food? When the bacteria are on the plates with more nutrients, what do you predict we will observe on the plates? So you're describing what you think will happen to the bacteria with no food, what will happen to the bacteria with food. Here are my images of the different levels of nutrients. So this first plate up top is 0% nutrients added. After 2 days, make some observations. 25% nutrients. Here is my bacteria growth. 50% nutrients, 75% nutrients, and 100% nutrients. Okay, so this is where you're going to want to grab a highlighter, and on your paper on page 13, I want you to highlight all of the colonies that you see on your paper. All right, this will help us be able to compare between the different colonies. All right, so using a highlighter, trace the streaks you can see on each plate, and then answer these two questions right in the video. Which plate has the most highlighting, and what was different about that plate? So if we look back at our plates, the 100% nutrients definitely had the most highlighting, the most growth, right? So 100% nutrients means I had a lot more bacteria growth. So the last step that you need to do is this slide right here. You're going to page 15 in your packet. And first, I want you to identify the effect. So this is a cause-effect relationship between the nutrients and the bacteria growth. So you're going to identify the effect by thinking about what happened or what was one finding from the investigation. So what was the effect? We saw some of the plates had limited bacteria growth, some of them had increased bacteria growth. So think about the effects and then identify the cause. So what did we do that produced the effect? Okay, so think about those two things and then see if you can fill in all three of these sentences. When there was blank, so that's the cause, the result was, tell me what happened. All right, if blank nutrients were used, more or less the same, then we would see, tell me what would happen to the amount of bacteria. And as the result of blank, we can see blank. So you're going to want to write the answer to all three of these sentences in complete sentences on your chart. And then I would like you to draw pictures to add to your explanation. So you could draw examples of a few of the plates, something that shows you understand the relationship between the amount of nutrition and the bacterial growth.